So what are you going to focus on this one? Many of it you have passed already. It's not hard really. It's not. But when we are coming to discuss how you are going to find unknown bacteria, we need to touch Burgess manual procedure. Burgess manual. What does it mean, that Burgess manual classification? I will touch that one. Before we go, I'm giving you this objective of this finding unknown bacteria. Why is it important? Well, you are going to stain it, the bacteria, and then by staining gram, spore, acid fast stain, you will find the bacteria, almost 90% of the bacteria, you know it, by those stains, and then you are going to do fermentative process. Why fermentative process? Because the one which you have done on 3 stage gram, spore, and acid fast, you need to accompany it, make it proof or make it perfect that analyze is going to be exact to prescribe medication for a patient. You see, for this reason, I'm bringing certain objectives as general to see it. What objectives are there? Establish working with unknown microorganism. Well, when you are making a streak or you are having a colony, from the colony, how you need to make it, gram stain, spore stain, acid fast stain, working with that one, it's a good example on microbiology laboratory. Okay. The other one, determine the optimum growth. That means those microorganisms grow in what temperature? We know most of them will grow around 37 degrees centigrade, which we call mesophils between 20 and 50 degrees centigrade. Well, that is good. And then the other ones which are not this much interested are hyperthermophile, which is greater than 80 degrees centigrade. So we are concerned on temperature also. Why? We are focusing more on temperature because those microorganisms like our temperature, and then they are going to be pathogenic on 37 degrees centigrade. You see, if it is coming horse or chicken, they can be around 40 degrees centigrade. But with us, it is around 37 degrees centigrade. Optimum temperature, we call it, you see. Now, the temperature is very important for the growth of bacteria. The bacteria which grows faster means the bacteria is pathogenic. Pathogenic bacteria grow faster, you see. That we are going to understand it on this process also. Understand how to isolate microorganism effectively. We know it from colony, how we are going to isolate bacteria, we know it, you see. So those ways we are going to touch. Be familiar with variety options of bacterial identifications. For example, you have studied on the lecture part 20, bacteria are having virulence factor like coagulase, staphylokinase, and then streptokinase as enzyme, and then leukocidin, hyaluronidase, and then those ones are very important enzymatic structure bacteria to grow in us. So for this reason, if it is staphylococcus, how do you differentiate? By coagulase, good. What about streptococcus or streptococci, streptokinase? That is one enzyme way. And then there is another way also. We are going to call it MSA, mannitol salt agar media. Staphylococcus can grow in, look at it, in a salty media. And then streptococcus, it doesn't grow in salty media. So you see, you have done it already by coagulase and then streptokinase identification. And then you have identified by gram stain already. And then by gram stain, spore stain, acid fast stain, you identify already. And then finally, you are going to use mannitol media, which is going to be staphylococcus growing on salt media, streptococcus, they don't grow. So those are characteristics which we are going to identify microorganisms. And then determine the gram reactions, microscopic morphology, how they look like. You see pinkish, bluish. You see? Motility. We are going to focus on motility, especially stabbing the gel. We are not going to focus more on that wet mount. No, not this much. We don't do in laboratory this much in wet mount. So cultural characteristics which we are going to grow in fermentative process. For example, what are you going to use for catalase test? We are using hydrogen peroxide. Hydrolyzing sugars, what do we use? We are going to use iodine. You see, those kind of things, all we are going to focus for identification of microorganism, which we are going to find unknown bacteria. That is very important on microbiology, really. So here, for me, because I was practicing so many years, it is easy, but for you, it is not easy, really. Let me go over for you. Look now. For example, this one, Burgess Manual. It is identifying microorganism. You have it in your lab manual, I think page 115, 16. Okay, now. So you can find it in Google around, anywhere else. If you find it, Burgess Manual class bacterial classification, it's good enough, really. So looking at here, I'm going to help you to understand this principle, how to find unknown bacteria. Look, very interesting. Many of them you have done it already. I'm going to support by somehow. Look, very interesting is gram-positive bacteria. They are rods and then cocci. Cocci, oval shape. Rods are spiral shape. Maybe some of them are like a rectangular shape. Okay, now. So rectangular shape, maybe a square shape. There are certain bacteria. And then the other are cocci. Cocci and then circular ones, oval shaped bacteria. So in gram-positive, there are rods and then cocci exist. You know it. Staphylococcus, streptococcus, cocci. And then bacillus group and then clostridium, they are rod-shaped bacteria. Look at how we need to find them. It is not hard, really. It's not. Looking at here, I'm going to lead you this way. Just see it. For example, rods are spore former. What are spore formers, which we have studied on a lecture part and lab part? These are bacillus and clostridium. Look now, bacillus and clostridium. You know it. You have done it, this one. So you know these bacillus and clostridium are spore formers. So on exam, I will give you, mix it up another bacteria with bacillus or clostridium, and then which of the following is spore former? You need to know bacillus or clostridium. Two of them you need to know on the exam. Okay, now. So now spore formers, we are having two. The other one is Sporolactobacillus, we didn't study. So don't worry on that one. So here bacillus and clostridium, remember, there are rods, spore former. Okay. What about the other one, non-spore former? Acid fast. Mycobacterium, is it a spore former? No. For example, I may say Mycobacterium is a spore former, true or false. You need to know it, you see now. Why you need to know? Because bacillus and clostridium are spore formers, and then mycobacterium is not a spore former. It is not given as a spore former here. So you can identify it, you see. Look now. So that is very important. Now, non-spore former is mycobacterium. The other part, these ones, we didn't study. So non-acid fast, we call them those ones, you see. So regulate or polymorphic, different shape they have, that you need to have general idea only. Now, you need to focus on those arrows which I need to give you. Okay, now. So we need rods, gram-positive, we finish. You see now. So how many of them you need to remember? 1, 2, 3 of them in gram-positive bacteria. Okay, now. In the other cocci group, look now, I need to go second one, which is going to explain you cocci group, gram-positive. In a cocci group, spore former exists, Sporosarcina, one bacteria. Okay, what about cocci, the other one, non-spore formers? Which ones? Okay, we are having Micrococcus. Did we learn in a class? We did use? No. The other one, which we have, Staphylococcus we used, cocci. Cocci is staphylococcus. And then spore formers. The other one is streptococcus. Did we study? Yes. Streptococcus, staphylococcus. Look now. So two of them you need to know from the cocci. You need to know them. You see, they are cocci. So these are gram-positive what? Cocci group. This one, you need to know it. Look now here. How many of them you need to know now? 1, here come 1, 2, and then 3. Okay, the other one, 3, and then you need to know, look now here, 4, 5 of them. We touch 5 of them, you need to know it. Okay, that is according to the Burgess manual, gram-positive bacteria, which are Rhodes and cocci, they are going to be classified. Okay, now we are coming to another part here now. Gram-negative one. Look now, in gram-negative one, it is not easy to memorize them. Look now. So here, what we need to know is gram-negative bacteria, which are Rhodes and then cocci. Okay, from Rhodes, which one we need to know? Aerobe, and then did we study Pseudomonas aeruginosa? No. No. So, but Pseudomonas is one of the Rhode bacteria, aerobic. Okay. What about another one? Facultative ones. Facultative one. Do we have Proteus vulgaris? Yes. You know it, Proteus PV. And then do you know Escherichia coli? Yes, both of them are gram-negative bacteria. Okay, if I'm going to say for you, I don't want to say it gram-negative or gram-positive bacteria, but I may say which of the following are gram-negative bacteria. So I'll mix with gram-positive bacteria and then ask you Proteus vulgaris. So Proteus vulgaris, you need to say it is gram-negative bacteria. Okay, for that reason, you need to know it. Proteus and then Escherichia coli. We said 5 already there, and then now 6 Escherichia coli, 7 Proteus vulgaris. Okay, now 7. The A is 1. The A is 1. Did we study Shigella? No. Calvicella? We studied, yes. How do we know? It is non-motile bacteria. Calvicella is non-motile. It is one of the bacteria which has very hard covering. And then in genetic information, chapter 7, we focus that one. How it is going to be attenuated, how it is going to be killed, and then the killed one with attenuated one work together, and then virulent bacteria it will be. This is the one which is causing pneumonia, you see. So Calvicella pneumonia is a gram-negative bacteria. Gram-negative bacteria, it has very hard covering, and then it doesn't have flagellum. So because of that, it is not motile. So you need to remember Calvicella is non-motile bacteria. Okay, now. So you need to remember. Is there another one? For example, cocci group. Okay, did we study Neisseria? No. Did we study Verticella? No. So you ignore it, this one. Look now. So here you are having Neisseria is the one which we said it coffee beans look like, and then it is gram-negative bacteria, which is going to cause you gonorrhea. You see, when we study sexually transmitted diseases, we touch, but the staining process, we didn't use it. You see, so this is how we come on gram-positive and gram-negative according to the Burgess Manual of Bacterial Classification. So we are coming to understand another part now. When we are going to study about motility, we are going to discuss about motility now. When we discuss about motility, we need to know this bacteria is motile or not. Okay, now, motile or not. Okay, how do we know it? We mentioned already, for example, Escherichia coli, Proteus vulgaris are motile bacteria. Calvicella is non-motile. Motile bacteria, they need to have flagellum. Okay, if it is monotrichous, polytrichous, amphitrichous, polytrichous, whatsoever, they are having flagellum. Because of flagellum, they move. As scientifically, when we say all bacteria can move, it says by theory. But we are going to know bacteria are motile because of its own flagellum. Okay, now we are going to identify them by weight mount, one, and then the other one is going to be on stabbing, on gel stabbing. Look, very interesting now. I am not going to ask you on the test about wet mount on a slide or concave slide or hanging drop type. I don't ask you, but I may ask you about stabbing. You take bacterial culture, any of them, and then pass it to the semi-solid media, and then you are going to see diffusion, and then diffuses in the media. If it diffuses, positive. If it doesn't diffuse, it's negative. So for this reason, motility is very important. In unknown bacteria, we do motility. You see, motility. So here now, what are you going to understand? Look here. In motile bacteria, we are having citrate positive. Look now, citrate positive. Are we going to know that today? Citrate positive and citrate negative? Yes. So these are bacteria which are going to have citrase, an enzyme. that a virulence factor break down the tissue and other artifact from our infecting host. So for this, we say citrase enzyme is very important. If this motile bacteria have citrase or not, we will focus on that one. I will show you the reaction on that. And then we are going to say lactose. And then there is enzyme lactase. Is lactase enzyme exist with that citrus positive bacteria? Okay, now, is it in citrus negative? Is it lactase enzyme exists or not? You see. So those are very important ones. MR, this is methylene red, precursor type of reaction also. I'm not going to mention this one. So here, I'm not going to ask you on the test. You see now, in this one, methylene red means we grow the culture, brose culture, and then on brose culture, we are going to drop 2 to 3 drops of methylene red. So when you are adding methylene red, the culture, that is brose culture, is going to change red, and that is positive. So we don't do that, you see. If you are going to do in laboratory, yes, you can do it. But here, I'm not going to pass that one. Okay, now, so lactose positive, lactase negative, we are going to focus on that one. I'm going to show you. And then we are having here now, when we say Citrobacter, is it lactose negative? What about Enterobacter? Is it citrus positive? Yes, Enterobacter. Like Escherichia coli. Okay, now, when we are saying Salmonella and then Erwina, we didn't focus it in our class, so we don't focus on this area. So Citrobacter, we don't. But Enterobacter, yes, we need to know those ones. Why? Because it is citrus positive and then lactose, here it says sugar, negative. You see, so those ones we are going to focus on our slides. In the next slides, I'm going to explain you. See, this is how we are going to see bacteria in Burgess manual classification, how you need to differentiate bacteria by motility. Look now, here we said motile bacteria and non-motile bacteria. Okay, non-motile bacteria, which one? Shigella. Is it non-motile bacteria? Yes. And then what about Proteus vulgaris? Is it motile bacteria? Yes. It is motile bacteria, but citrate negative. You see, it's a very complex science. Look now. It's a very hard mathematics. It is really, it is very complicated design. It's not easy on this one. To ask you on the test event, it is not easy. It's very complicated with this one. But have an idea now, and then I will go further what you need to understand. So in gram, we focused already. On motility, we focused already. After that, we are going to do is fermentation process. Fermentation process. Okay, now, here, look now. Because of the gram stains, spore stain, acid fast stain, we know something. What are you going to know? So these ones already in gram stain, we are going to change gram positive and gram negative, and then in spore forming and non-spore forming, and then when we are coming to acid fast, we are going to say acid fast and non-acid fast. As generally, acid fast bacteria are gram positive bacteria. You know it. In the past, we were using like that. Even in certain literature, all bacteria classified by two techniques, by two cell wall structure, gram positive and gram negative. And that acid fast bacteria, Mycobacterium, Semigmatis, and the Mycotuberculose are classified as gram positive bacteria. So if you are going to say acid fast bacteria or gram positive bacteria, Mycobacterium, you are welcome. It is right. You are not wrong. You see, in the past, it was classified as gram-positive bacteria. When we want to be more specific, it is going to be gram acid-fast bacteria. So this is Burgess manual. Look now, this is Burgess manual, how gram-positive bacteria as rods, and then this one as cocci, and then as we come motility, gram-negative bacteria, and then rods and cocci, and then we come to motility. Okay, now, what are we going to know on gram positive and gram negative? Look now, and then including acid and the spore. Here, the unknown bacteria, when they do, my students, especially face-to-face, what they do, look now, they are going to do gram stain, and then spore stain, and then acid fast stain. Three of them they do. Look now, this is one activity. We don't have a lecture. Only they do this, three of them. And then if the students are doing very well, they make it almost 90% of the staining procedure and now bacteria, they know it by this one. If they have done it good. How they do it? Look now, very interesting. They make it, I give them in a tube, a bacterial culture, and then they pass it to make it a streak. And on when they do streaks, they are going to write their initial and then their roster number. Roster number. Okay. So nobody is going to touch this one. Only one student is going to do. And then now look, after this, the students are doing gram stain, spore stain, acid fast stain. They take a picture of the slide. You see, even on the slide, you know this bacteria is spore forming. Look now here, it shows. And then this one, reddish, acid fast. Here it's not clear, but it is bluish color. So for this reason, this is a technique students are going to submit. There is 5-page information the students needs to submit at the end. That all process I will teach you now. Look now. So look now. Here they have done it, and then the result. Look now. The result. This is gram-positive bacteria. They said gram-positive is purple, gram-negative is pink. Okay. Now here they come, spore, green, and then vegetative, pink. And then when they come acid fast, they say acid fast is red, the other one is blue, because they use methylene blue. Well, look now. So this one, how they have done it, and then this is the one they result. Almost 85, 90%. 90% they know there are no bacteria with this one. Look now, the student, when they do this one, if they finish it, they go home. Really, they go home. And then the ones who have done mistake, they are not sure, and then they need to come the second day and then do the same thing. Gram stain, spore, and then acid fast stain. The ones that have done already, they don't need to come to my class, you see, because they know it already. So that is a good technique we practice in, we were practicing in the class. If you are going to see, here you are going to see, look now, spore forming bacteria. Do you see that? Vegetative is red, spore is there. So they know it. Their bacteria is already spore forming bacteria. Which one? It can be Clostridium or Bacillus. That's all. You see, the very easy technique. They know it. You see. After that, they want to prove it by acid fast system. You see, very beautiful. Really, this is very beautiful technique by which is done by the student. My student, I've done it. Okay, now look here, this one. So gram stain, you know it. So in gram stain, crystal virate, gram iodine, decolorizer, and safranin, 4 of them you need to remember. And that we focus already. And then these colors, you need to know them. How they need to appear. On when they are going to make it gram positive and gram negative bacteria, even acid fast bacteria, to make it, the cell wall is very important. Gram-positive bacteria and acid-fast cell wall bacteria, acid-fast are having the same peptidoglycan amount. This is higher concentration of peptidoglycan. Which one? Acid-fast and then gram-positive bacteria, 1. Secondly, which you need to know is, look now, here, gram-negative bacteria, they have a smaller peptidoglycan. Okay, a smaller peptidoglycan, 2. Great, small, gram-negative. Okay, now, in gram-negative bacteria, there are another things which we need to know. 3 things, generally. 1, it is going to be smaller peptidoglycan comparing to gram-positive. 1. Secondly, they have a plasma, this membrane, outer membrane we call it. Outer membrane we call it. 2. This outer membrane protein, outer membrane is going to contain outer membrane protein and lipopolysaccharide. 2 of them. Look now. So you need to know 3 things in gram-negative bacteria. Which one? Peptidoglycan is small comparing to gram-positive, and then you are having membrane. This membrane is going to contain membrane protein, and it is going to contain also a lipopolysaccharide, LPS. You need to know it. If it is gram-positive or gram-negative, it is depending on the cell wall structure, not in the capsule, not on the plasma membrane or cytoplasmic membrane. No. So it is a cell wall structure. generally. Okay, now here you know it, spore forming, the two chemicals you need to know. So you are need to know malachite and then safrani. Now then, and then what I may do is I'm going to give you this kind of picture, and then I will magnify it more, and then I will show it by arrow, and then give you multiple choice. Is it vegetative stage or spore stage? Or what kind of bacteria can form a spore? That is bacillus and then clostridium. You need to know it on the test I'm going to ask you. For example, this one is very good one, but because of endospore is written here, I'm not going to. I'm not. So I will ask you from this one, which is not showing the name. If I want, I will cut it and then make it, I will magnify it also. So for this reason, be ready on that one. It's not hard one. So you may focus on this. Okay, now, when you are coming, look now, this is spore forming. Without heat, we can make it. Or with heat, also, we can make it. With heat also. This is heat. We are using heat also. We can make it. But the problem is only it may take longer time. We do. So for this reason, heat is important. And then without heat also, we can stain spore. But Schaeffler method, we are going to use heat method. Schaeffler method. Okay, now, so that you don't need to worry about Schaeffler's name. Okay, here when we come, you need to understand about acid fast. Carbolfuchsin, you need to know it, and then acid alcohol, and then methylene blue. Know it. Most of the time, students, they say acid instead of acid alcohol. Don't make a mistake on that. You need to make it acid alcohol. You see, that you need to know it. So what color it will be on acid fast, you need to remember it. So the three of them, you need to know carbonfuchsin, and then acid alcohol and methylene blue. Those ones you need to know. At least brilliant green, if you are going to use, it will work. You see, it will work also. But according to the given, methylene blue is good one also. Look now here, you are going to find it as final. You are doing the gram stain, you are going to do spore stain, you are going to do acid fast stain. Look now here, you are going to do them, and then finally you know it, and then you guess 85 to 90% your bacteria is. Almost by seeing this one, the second one, you'll say, my bacteria is a spore former. But are you going to check, is it bacillus or clostridium? That is the important one. So for this reason, you need to do fermentation process. Okay, now, so we need to go further to that one. Okay, motility we discussed already. According to Burgess manual, we need to focus on agar stabbing method. This is semi-solid media. It is not solid media, semi-solid media. And then by stabbing, you are going to see this diffuse, and then triple phenol tetrazolium chloride exists or not, you don't need to worry. But you need to, if I give you in multiple choice, this one, or I may give you this one. This is the perfect one, especially. So if I am going to give you, which of the, I will say A and then B, which of the following is non-motile and then which is motile? You need to know it. It's not hard. Okay, now, this is only motile area. You remember that one. Okay, about motility, we are going to focus more also in the future. So here, for example, which we are going to do motility and then bacterial enumeration. For example, these ones are going to be 1 day for laboratory activity students are going to do. Motility and enumeration of bacteria by colony forming or spectrophotometer, you see. But we are going to finish it in 1 day, this one. We are going to work together today when we are going to find unknown bacteria. So we don't spend more time. We don't have another time which we are going to practice laboratory. This is only information which I'm going to give you how to find unknown bacteria. Look now here. Are we going to do this one? Well, we are not going to do dilution, but we are going to do, look now, there are bacteria here. I made it colonies. Colonies are reddish, pinkish, are here on this surface. Now, they are having a live bacteria. Why is it focusing on live bacteria? A live bacteria, they grow, they don't die. So when we say colony, you need to know the bacteria is, look now, the bacteria is colony forming, means the bacteria is alive. Okay, that you need to know it. So here about more bacteria, when you go further, they are going to be less and then less. It doesn't matter about the dilution, but there are colonies there. Colonies are product of alive bacteria, not dead bacteria. You need to know it. I can ask you by true and then false question also. So now, colony forming, CFU, means alive bacteria. Okay, when we are coming to a spectrophotometer, it is going to count more bacteria in the form an antigen or bacterial cell themselves. Why? Because it is going to count dead and then alive. Is it hard? No. So spectrophotometer can read dead and then alive. So that is the only thing two you need to know. One is alive, the other one is dead and alive. And then which one is having more bacteria to enumerate? It is going to be a spectrophotometer. That you need to know. Very beautiful with that one. Now, when we are coming to understand about disinfectant and then antiseptics, you need to know this one. For example, I'm going to ask you this one, not on this one, not only making these discs and then inhibition. I may not ask you. I may not ask you. Even I may not ask you the chemical itself. Sodium hydroxide, Lysol, or formalin. I will not ask you. So I don't want you to memorize it, but I will give you a scenario. What kind of scenario I may ask you? Okay, you are working in laboratory. There is bacterial culture in your hand, somehow touch you and then fall from your hand, and then it split on the table. What measure will you take around the table? Around the table, you are going to use a disinfectant, not antiseptic. Why? Because the table is not living. What about if I'm asking you, you touch it and then the bacteria split on your body? Now I'm not going to mention disinfectant, I'm going to mention it antiseptic. Why? Antiseptics are going to be used on your body surfaces. That kind of scenario, you need to know it. You see, that is what generally you need to know. So inhibition test, I'm not going to ask you. I will not. Measuring whatsoever, chemicals name, no. You need to have disinfectant are used for non-living objects, and then antiseptic used for the body surfaces. That is enough for me. You see. So that is all what we need to do it. Also, we need to understand it. So about when antiseptic, this kind of inhibition test whatsoever, I'm not. But you need to understand it that antiseptics are going to be used on body surfaces. That's all. Okay, now susceptibility test on antibiotic. The students are given antibiotic, which are going to have 50 milligram, 20 milligram, streptomycin, penicillin, erythromycin. So here, what you need to know is we are going to do inhibition test with uh, antibiotics. The disc are having already certain milligram of antibiotic. So what you need to do is you are going to pass bacteria. Students will do it in my class. They do it surface all they cover with bacteria, make a strict completely. And then here also they make it completely. And then why will they make all completely? Because we need to measure the inhibition test, how it is sensitive, susceptibility of that antibiotic, how it is going to inhibit the growth of microorganisms. That's why we need to spread it completely. You see, we don't want it transparent. We need to deeply we make it. Look, this one is very beautiful. This is not. This is transparent a little bit. So this is the best one. Look now, covered all surface almost. So here you are saying this is broad spectrum, this is broad spectrum. Now this is resistant. Do you see that? That is inhibition. And then susceptibility. Doctor is not going to prescribe medication like this one. If it is streptomycin is resistant, the doctor will not. But may prescribe this one or this one. That means inhibition test, we call it. Susceptibility test. So the doctor is prescribing you that kind of antibiotic, maybe to take it 3 times a day if it's acute case. If it is not acute case, maybe 2 times a day. You see? So this way to forecast, you need to know the inhibition test, how it works. You see? So for this reason, sensibility of antibiotic, we are going to try like this. So when you are going to find it is a staphylococcus, a streptococcus, or bacillus group, when you are going to find it by gram staining, or by spore forming, it is, you found it is bacillus, and then that bacillus, how is it sensitive for this kind of antibiotics? Okay, that is why we need to do susceptibility. You see, it's a very interesting subject, really. Now, so when we are coming, agglutination test. Look now, agglutination test, you have done it, this one. You can do blood also. You take a blood. If you have blood, you are going to use blood as an antigen. You are going to use antibody, and then antigen, antibody, antigen of the blood, and then antibody from the outside. You are going to make it together, and then you can read coagulation. Make some kind of granules. You see, granules, 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 granules. You see, those granules make it positive. That is agglutination test. It makes agglutinant, we call it agglutinant. These are bunch of bacteria are joining together, making some kind of sediment, precipitate on the slide. You see, so we call agglutinant. And this one, it is negative. There is no agglutination. It is homogeneous. Homogeneous. From the beginning up to the last, this liquid is homogeneous, equal. In this one, no homogeneous, no homogeneous, no homogeneous, no. You see, there are granules. So for this reason, agglutination is positive. Is it good? Yes. Yes, it is good, because antigen is going to recognize antibody. Aha. So the doctor is saying, oh, this bacteria is bacillus, maybe it is gram-positive bacteria. And then when we are going to do sensitivity, the erythromycin is going to be inhibit very high, or vancomycin or penicillin is inhibiting this one. And then let's see immunologically how it works. Immunologically, they analyze it. They take your blood and then make it antibody, which is known, make it antigen antibody. Okay, then if you don't want it, then you centrifuge, take an antigen antibody, you can make it. You see, so the blood is going to have antibody also. Okay, what you need to do now, an antigen you take and then mix it with antibody from here in the blood. So immunoglobulin A, immunoglobulin G, immunoglobulin D, immunoglobulin B, immunoglobulin M are here on this one in a blood. Because if you are going to centrifuge the blood, you are going to have this plasma. Okay, the plasma you are going to centrifuge, or higher centrifugation you do the blood itself, you are going to find the serum. Okay, if you are going to leave it in a room temperature, yes, you can find plasma, but the plasma is centrifuged, you are going to find antibodies, immunoglobulin groups. Okay, now, so for this reason, antibody exists already in this one, in the blood. So if you are having antigen now, then you send it antigen to this one, to this one, and to this one, and then this one. The antigen antibody connection is making positive reaction, and we call agglutination positive. You see, it's very beautiful. And then you have been doing this one in a blood typing on physiology. A, B, AB, and O. You see, you are having an A, B, and then you are going to use antibodies against those ones, and then you are making a reaction. Agglutination make it, it says that group is A or B. Okay, so if it is going to be A positive and then B positive, it is A, B blood type. If it is no A positive, it is no B positive, then it is O group of blood, you call it. So there is no agglutination, you see. For this reason, the background is the same in physiology too. So agglutination is very important, really. I like that one. And then this is how we need to focus. Look now, agglutination test. See if it works.
Agglutination assays have been used for decades as a simple method to detect antigenic substances in biological samples. The purpose of this video is to explain how this method works in practice and to expose its limitations. The agglutination assay uses tiny particles, most often latex beads. The beads are coated with a specific antibody against the antigen that you would like to detect. The test is usually performed on a card or glass or plastic slide, often one with a black surface. First, you add a suspension of the coated latex beads to each of the 3 encircled areas on the slide. Note that the suspension is concentrated enough to produce a milky appearance on the background. Now you add a few drops of the unknown sample that you're interested in testing, but you will also need to use one circled area for a negative control solution that contains no antigen, and another for a positive control solution that contains the antigen of interest. Next, the slide is gently rocked or swirled to mix the beads with the test solutions, and the samples containing the antigen of interest will begin to agglutinate the beads. This will produce the appearance of visible clumps, and the solution itself will turn from milky in appearance to clear and transparent. This transition should occur in the circle with the positive control. If the antigen is present in the unknown sample, then it will form clumps. The negative control circle should remain unclumped and opaque. Recall that the latex beads are coated with a specific antibody so that each bead can bind to numerous antigens. For agglutination to work, the antigen of interest must also be able to bind to multiple beads.
Okay, in this one, you have seen it, how the reaction is going on. So this is very dedicated laboratory activity. If you don't have that beads are having certain structure of adhesive character with antigen and then antibody, you can take a blood itself. For example, you take from the client a blood, and then you have antibody already, antibody, and then that blood which you join together, make it on the smear in the clinical settings. Without having deep beads, you can do it also. Yeah, you are going to get it also. After that, if it is positive, then you are going to send that blood in laboratory to verify it. Final. So because you have done it in a clinic, it is not enough. You need to identify what kind of bacteria, and then how it is, and then all structure you need to be identified with that one. The beads being embedded with certain kind of antigenic structure is not always applicable. You can do it. In veterinary, for example, we go on the field, we are having a slide, and then the antigen, we make it those ones on the field. Without any beads, we can do it. But we are about 95% perfect on that. But if you are having embedded type in pure research, you do it, then you need it, you see. So that is very interesting on agglutination. I have it here now, gel immunodiffusion one. It is like agglutination also. Why? Here in gel immunodiffusion, immunological, we are using antibodies here now. Look now. In one side, there is antigen. In the other one, it is antibody. Look now. Here, antibody, and then antigen, and then making precipitation between. Very beautiful. Isn't it interesting? Very interesting. Precipitation. So they note with this one, there is no. So this antigen, if this is antigen and this is antibody, this antibody is not recognizing this one. If it is an antigen also, it doesn't recognize it. Very good. So for this reason, we call it gel immunodiffusion, we call it. The technique here, I'm going to explain you that also. Look at this one. For example, these all can be antibodies or can be all antigens. And then this is antibody. So this antibody is going to recognize all antigen. Or this is antigen, and then these are all antibodies recognizing, and then they are making precipitation. Isn't it interesting? Very beautiful. Look. So that is how we are saying immunodiffusion or gel immunodiffusion. Well, this kind in so many ways, really, so many ways. I'm going to this one in our lecture on chapter 22, serological reactions. Serological reaction, when we say it is a study of serum. So serum is containing antibody. This antibody can be used in different ways. ELISA, we call it enzyme-linked immunoabsorbent assay. And then this complement fixation test, CFT, we call it, antibody. And then we are going to use counter immunoelectrophoresis. Counter immunoelectrophoresis. It is going to be like this, but it is going to be like this, but we are using electric current in a tampon solution. you see. So that's all we are going to touch in the future, how immunodiffusion is going to work also in a lecture part in serology. It's a very interesting topic, you see, very interesting. So Western blot techniques whatsoever, how they make precipitation lines, patterning between proteins, linings, that we are going to touch on serology chapter 22. Well, this is how we are saying gel immunodiffusion, precipitant. Aha, precipitation sign. Okay, now, how it is going to explain you this gel immunodiffusion?
Oh, they're still on board work with you. Did you hear that? It sounds good now. Yeah.
Outer-Looney double diffusion for antigen antibody patterns. Objective: to illustrate antigen antibody interaction and learn the technique of Outer-Looney double diffusion. The key reaction of immunology and immune defense is the interaction of antibodies and antigens. The specificity of antigen-antibody interactions has led to the development of a variety of immunologic assays which can be used to detect the presence of either antibody or antigen. In Outer-Looney double diffusion, both antigen and antibody are allowed to diffuse into the gel. Antigens from different species are loaded into two wells, and the known antibody is loaded in a third well located between and slightly below the antigen wells to form a triangle. Depending on the similarity between the antigens, different geometrical patterns are produced between the antigen and antiserum wells. The patterns of lines that form can be interpreted to determine whether the antigens are same or different. Materials required: agarose, 1x assay buffer, antiserum, test antigens, glass slide, gel punch with syringe, template, incubator, conical flask, measuring cylinder, alcohol, distilled water, micropipette and pipette tip, Petri plate, cotton. Procedure: prepare 15 ml of 1.2% agarose in 1x assay buffer by boiling to dissolve the agarose completely. Cool the solution to 55 to 60 degrees Celsius. Wipe the glass slide with alcohol to make it grease-free for even spreading of agarose. Pour 4 ml onto the glass slide placed on a horizontal surface. Allow the gel to set for 30 minutes. Punch wells by keeping the glass plate on the template. Label the wells on the opposite side of the glass slide. Fill the lower well with 10 microliter of antiserum and the upper two wells with 10 microliter each of antigen 1 and 2. Keep the glass plate in a moist chamber overnight at 37 degrees Celsius. After incubation, observe for opaque precipitin lines between the antigen and antisera wells. Results: pattern of identity A. In this pattern, the antibodies in the antiserum react with both the antigens, resulting in a smooth line of precipitate. The antibodies cannot distinguish between the two antigens, that is, the two antigens are immunologically identical. Pattern of partial identity B. In this pattern of partial identity, the antibodies in the antiserum react more with one of the antigens than the other. The spur is thought to result from the determinants present in one antigen but lacking in the other antigen. Pattern of known identity C. In the pattern of known identity, none of the antibodies in the antiserum react with the antigenic determinants that may be present in both the antigens. That is, the two antigens are immunologically unrelated as far as that antiserum is concerned.
This is one of the gelatin hydrolysis. Gelatin is a protein. It looks like a jello. Okay, now. So here it's a very simple technique. It's very easy also. I may give you on the test, for example, this one and this one. And then I remove this letter or whatsoever. Look now. Here, very interesting thing which we need to know is gelatin hydrolysis. It means certain bacteria bacteria are having gelatinase enzyme, gelatinase. That gelatinase enzyme is going to break it down, gelatin. So for this reason, when you are going to keep the bacteria in a room temperature, and then even 68 degree Fahrenheit or 22 degrees centigrade, if you are leaving it in room temperature around 18 or 20 degrees centigrade, you are leaving it in a room temperature, because when the bacteria grows, it is going to melt it because it has gelatinase. If the bacteria does not have gelatinase, this is not going to melt. So for this reason, we are saying gelatinase negative and gelatinase positive. You see now. So there is explanation for you better than I do here.
Okay, in the last video, I forgot to mention one thing that I wanted to just do right now. So this is going to be a little add-on. So it's going to be posted separately, but just go look. It'll be there with the rest of the videos. This one is the gelatin results. So I told you for all of the plates that you're looking for that zone of clearing as a positive result. The starch you had added the iodine, but the rest of them were just the plate itself. I forgot to mention the gelatin. So if you recall Jell-O, okay, so think of gelatin as being similar to Jell-O in consistency, maybe a little bit harder than Jell-O, but when you let Jell-O sit at room temperature, it will melt, okay? Gelatin is the same way, okay? So these will be sitting in the incubator at 37 degrees and they're obviously gonna melt. Well, the result that you're looking for is, does it solidify or does it remain a liquid when cooled? So what we're gonna do is, there are a couple ways to do this. We could put them in an ice bath for 30 minutes or we could put them in the fridge. So what I'm gonna do is I'm gonna take your gelatin out of the fridge, out of the incubator, and put them in the fridge at the beginning of class so that you'll have a result by the time you go to do it, okay? So if the gelatin, once refrigerated, remains a solid, so I'm turning this and there's no liquid coming, okay? If it remains a solid, that is a negative result because the bacteria did not have the gelatinase to break it down. If once refrigerated it remains a liquid, that's a positive result because the gelatinase has broken down the gelatin in the media. Okay, so for the gelatin tubes, we're gonna put them in the refrigerator, wait, and then you're going to look to see if it remains a solid or if it becomes a solid in the fridge or if it remains a liquid. The other thing about this test, the gelatinase, is it can take up to 7 days for that result to happen. So what you're gonna do is you're gonna look at them on the second day after 48 hours, then you're gonna put the gelatin back in the incubator. The cultures in the gelatin, you're gonna put them back in the incubator. The following week, you're gonna look at them at a second time, and you're gonna see if it has slow or fast. So if it happened within 2 days, or it may take 7 days for that result. So this lab will actually extend an extra day just because of the gelatin tubes.
Now, this is agglutination, which I told you already about the blood and then antibodies. Join together, agglutinant. You see? So this is not agglutination. This is homogenized. So here is agglutination negative, here is agglutination positive. So those are how we are going to do in blood also. It's very interesting. When we are going to understand also, we are coming to understand fermentation test. Already we say carbohydrate fermentation. Here now, please focus on this one. I'm going to give you some idea. I'm going to ask you on the test this one. Please remember it. Any fermentation process we do, we are going to expect acidity, production of carbon dioxide, and then hydrogen gas. Yeah, don't forget that. You see now. So when you are going to have some water and then add sugar, close it, and then keep it for about 24, 48 hours, fermentation bumps up. If it is plastic one, it bumps up. Why? Because there is carbon dioxide producing also gas, hydrogen. And then the media is going to change the color. Acid, you remember when we were in high school even, when we say acid, red, acid D final, red D. And then if it is base, it is blue, you see. So when fermentation process goes, it is going to change acid. You remember when we were talking about metabolism of bacteria on chapter 6 also, in physiology is metabolism, anabolism, and catabolism. When you mention, then in fermentation process, the carbohydrate break down, and then they are going to change pyruvic acid in the process of hydrolysis. Look, that carbohydrate breaking down, they are making acid, you see. So for this reason, acidity will be created by fermentation process. Don't forget it. And then fermentation without carbon dioxide never can be created. So you put it, sugar, and this one, leave it open. No fermentation goes. Carbon dioxide must be there, you see. So you need to firmly close it, fermentation process. And then it produces hydrogen molecule too. Here you are having, for example, this is tryptose broth, for example, adding certain chemical, color changing here as a control. And then this one, look now, it is going to grow. Bacteria is changing the color. You see, it is acidic. And then it forms a gas also. Why? How do you know it? Look now, this tube itself, it makes meniscus at the bottom. Meniscus. That meniscus positivity for gas formation. And also empty at the top, gas formation, you see now. In this one, it doesn't form a gas, you see. In this one, it doesn't. But is it change acidity? Yes, you see. So for this reason, it is early stage. When it is stay more longer, it is going to show you meniscus also, you see now. So for this reason, for your understanding, fermentation process is going to give you carbon dioxide, acidity change, and then hydrogen gas. So here it is going to explain you some more.
Okay guys, so let's talk about how to read carbohydrate fermentation test. So carbohydrates include all of the sugar tests that you could be doing, such as sucrose, lactose, maltose, or mannitol. All of the tests are individual sugars, however, their reactions would be read exactly the same way. For example, in this tube, you'll see that this tube represents the uninoculated media. The next tube represents a negative result, in meaning that the organism does not ferment this particular carbohydrate. The next tube indicates a positive result, and there is no gas production here. Notice the tube inside and out, and including inside the Durham tube, is completely yellow. The next tube is a positive result with gas production. Gas production can be seen by the meniscus line in the Durham tube being much reduced from the outer meniscus line of the greater tube. So notice this empty space inside the Durham tube would represent gas production. The next tube is known as positive in the Durham tube. So in this tube, you'll notice a hot pink coloration outside with a yellow inside of the Durham tube. This means that the bacteria have other than oxidative or fermentative means of reducing carbohydrate, and so the outside of the tube would be an alkaline while the inside of the tube would produce acid.
The other reaction is going to be citrate utilization. Citrate, you see, the bacteria is having, if the bacteria is pathogenic, and then it is going to use its own enzyme citrase. Citrase, it is going to break it down or changing the color. Look now how here it is. For example, this one is negative. It doesn't have citrase. This one has citrase. How do you know it? Because it changes color to Persian blue, we call it, Persian blue. Means there is an enzyme citrase. You see, this test distinguish different genes of Enterobacteriaceae, like Escherichia coli, Proteus vulgaris, you see. So because it has citrase enzyme. Look now. So here is positive, here is positive. In exam, what I may show is, I will show you this one. This test is showing you, hey, citrate utilization, hydrolyze utilization, and I'm going to say glycolysis utilization. You need to know it. You see, the color. This color is citrase, and then it changed to the blue, and we call it Persian blue color. So that is very important. Now we can put it on the plate also. Look here, it is going to explain you in another way here. For example, here you are going to have, now you look here, blue, positive, and here, negative. And then on the slide, here is blue, and then here is negative. Here is negative, here is blue. So citrase exists on citrate agar. You see, the microorganism is using an enzyme and then break down citrase, which is going to be in the media. So that is one thing which we need to understand it. Now, catalase test is very interesting also. We can make it in different way also, this one. Look here. Catalase is very important. You remember when we say catalase, coagulase. Look now, we are saying staphylococcus or staphylococci is going to have a coagulase enzyme. That is a virulence factor of that microorganism. Look here, very interesting thing is that microorganism is gram positive, and then enzyme is coagulase, or we can call it staphylokinase. If it is streptococcus, we say streptokinase. It is an enzyme for streptococcus. Okay, one means that. And then the second one is catalase. Look now. In a catalase, you are going to have to differentiate this bacteria, staphylococcus, and then streptococcus. I am going to bring you another, the third one also. The third one, you see, coagulase, streptokinase, one way. And then now we are coming staphylokinase, and then streptokinase. And then when we are using catalase, they are reacting positive or not. Look now, in this one, staphylococcus is having an enzyme, you see, an enzyme. And then when we are going to add hydrogen peroxide, H2O2, when we add here bacteria on solid media, it can be, you can put it on a slide, and then we want to drop 1 or 2 drops of hydrogen peroxide, and then it dissolves and then make it bubbles. Those bubbles are called water and oxygen. Oxygen is a one which makes bubble. So for this reason, hydrogen peroxide by catalase is broken down to water and then oxygen. Look now. So which bacteria is it now? It's staphylococcus. You differentiated staphylococcus and streptococcus. This is a non-bacterial finding method. Very beautiful, really, very beautiful. And then if it is there, another explanation exists. This is very long process. I'm not going to mention it. It goes very far on this one. Now, this is molecular biology base. We understand it differently, really. There are so many problems on this lab, on this technique. If it is not very long, let's make it short. Yeah, 3 minutes.
First, I'd like you to read the description for this experiment. You can see inside are the steps. First, we're going to drop hydrogen peroxide on the slide. Then we're going to pick an isolated colony from our plate. And we're going to mix that colony, that bacteria, with the hydrogen peroxide and look for bubbles.
Yep. And I'm going to give you more here.
All right, let's get started.
Look now here.
Here's our setup. You can see we've got all the different components we talked about. So the first thing we needed to do, remember, is add hydrogen peroxide to our slides. So there's the first one. Here's the second one. Next thing we want to do is take a colony from one of these two and add it to the hydrogen peroxide. So let's see if we can get an isolated colony here. Oop, missed it. Let's try again. There we go. And we'll put it in this hydrogen peroxide. Woo, stir, stir, stir. And pay attention to the bubbles. What do the bubbles mean? That means we have a positive reaction. Remember, we want to make sure we dispose of this in the correct biohazard disposal container. Okay, so Staphylococcus aureus is catalase positive. Let's try Streptococcus pyogenes. Oops. Grab a stick. And we need to make sure we get an isolated colony. This one looks good. We'll swirl it around in the hydrogen peroxide. And no bubbles. That means it's negative. All right, so Streptococcus pyogenes is negative. All right, I'm going to dispose of this.
That is how generally we read it now. So when we come, catalase test, look, catalase, we understood it by that way. And we are coming to starch hydrolysis, glucose breakdown. Starch, it is in your mouth also, how it is going to broken down carbohydrates. You see, because of the amylase enzyme in our mouth, parotid, submandibular, sublingual glands are producing saliva. That saliva is injected to our oral cavity. In the oral cavity, we are having carbohydrates, and then those carbohydrates are broken into soluble stage by the presence of amylase. Now, here you are going to understand now, look now, you are going to add what in this one? The most important thing which I want you to know is on starch hydrolysis, what do you add? Okay, now, this is a media. It can be in solid medium. It can be on a plate. On a plate, you are going to have 1 or 2 drops of iodine solution. Iodine solution. Look, iodine you add, it is going to change it brownish, dark brownish color. In this one, no. It is start, but it's not. Look now. In this one, here, this is the media, and then you add it. This iodine, it changed brownish color. And then you add it, it didn't change on this one. Look now. So for this reason, this is positive for hydrolysis. This is negative for hydrolysis. Look now, very interesting. So for this reason, we are going to do starch analysis by this way. So I have it, this technique, look now here. Very interesting on this technique. For example, we are going to hydrolyze, that means on agar media, we are going to use iodine, and then if the bacteria is going to be hydrolyzed by the presence of amylase, then it is going to break it down. That is one thing we need to know. In this one, it is going to show you how it goes.
Some bacteria release the extracellular enzyme amylase, which digests starch.
Sorry.
Some bacteria release the extracellular enzyme amylase, which digests starch. And if bacteria are spread onto starch plates, which have starch incorporated into the agar, and incubated for several days, those that make the enzyme amylase will digest the starch in an area around the bacterial colonies. When the plates are then flooded with iodine, this iodine will convert the starch to a blue-black color. If bacteria produce amylase, the area around the bacteria will not turn blue-black because the starch is no longer present. It has been digested, and it will be clear instead. So a clear zone around the bacteria indicates that the bacteria make the enzyme amylase. They are amylase positive.
Yeah, that is a good one. And then here you are going to use, if you are going to mention the other one, spirit blue. This is fermentation technique also. Fermentation technique. Spirit blue is a test which we are going to find if there is lipase or fat exists there. Lipase, lipid, fat. Okay, now, so what you need to do is, look now, here, for example, you pass it on the plate like this, the bacterial culture, and then the other one has this one, and then because it is going to ferment, this media to change it to spirit blue, and then that means the enzyme is, that media is going to break down the lipase, the lipid on it. So that is how we are saying spirit blue. Be sure when we are going to talk about, when we were saying another reactions which the color Persian, the green one, is going to change blue, you see, that you need to make it different from the other one, you see. So that lipase is virulence factor of that microorganism, which is emulsifying certain fats in a media also. So when we are going to see, look now, here you are having the media, agar plate having a media, and then here it is going to change bluish, and that bacteria has lipase breaking down structure. That's why it is going to change the color. Very interesting. So that is spirit blue agar plate, we call it. So when we are coming, another one, urease test. Urease test is to analyze if that media is, that sample which we took has Escherichia coli or Helicobacter or another bacteria, you know. So here, very interesting thing is, this urea is having an enzyme urease. So any bacteria which is going to grow on urine, it is going to use urease enzyme, and then it is going to change the color from this one to purplish color or cherries color we call it, purplish color, you see. It's very interesting. Now, here it is positive for urease. That means like a bacteria, Helicobacter, or we call it Campylobacter pylori or Campylobacter fetus. This is gram-negative bacteria, which is going to attack especially around the duodenal area, duodenal ulcer causing, or at the bottom of the stomach where alkaline media is. So it is going to use this kind of neutralizing the bile, which is going to release from liver, and then it neutralize it, and then it may stay around the duodenal area and then in the stomach, bottom of the stomach, and then it is going to do certain infection process, non-healing wound. Mostly people are going to be infected with this are between 24 and 64 years old. It's a very interesting bacteria, gram-negative bacteria. So it may use urease enzyme. And then when you are going to take a urine, you don't need to take another sample. You don't need to go ultrasound for that. So you will find it. Biopsy you can do, yes. By biopsy you can analyze it. Histology you do it, and then you culture it to find the bacteria, yes. But the easy way is take your urine, analyze it, if there is Helicobacter pylori or not. You see, this is mostly water contaminant bacteria. Some kind of water you drink, some kind of food, which are the fresh fruits, which are contaminated with bacteria, and then you can be infected. We call it Helicobacter because it looks like helicopter itself on the shape. It doesn't have uniform shape, but we call it Helicobacter. Now it is going to change it, the media, into look in the cherries or purplish color. You see, so it's a very interesting bacteria, really. So when we are coming to understand, I am not going to use that video. Now, litmus milk. We are having about 2 or 3 of them. We finish almost. Now, litmus milk fermentation. It is very important. It's a fermentation process also. Litmus milk. Look, very interesting this one. I'm going to open and then you are going to see the video also. Very interesting. Why is it very interesting? This litmus one. Because we are going to find the cheese. We are going to making cheese and we are making yogurt, you see, because of fermentation process. You see, it's very interesting. You see here now, you are going to find the milk as it is, and then the milk natural, and then this is milk alkaline, bluish, alkaline at the bottom, and then this is going to have coagulation of proteins inside. Protein is making yogurt. Look now, here also coagulation, and then we call peptonization, means breaking out of the milk protein. Look, peptones, peptonase enzyme is breaking down this one to peptone proteins. So is this protein exist in cheese? Yes. Is it exist in yogurt? Yes. So these all are fermentation process. Look now, here is alkaline, and then here not alkaline. Here acidity. Look now, acidic. Red color, almost acidic. At the top, it is alkaline. Some kind of breaking down peptonization you can expect also. Look now. And then this granulation of the milk to make a cheese, yogurt, you see. So this one, if you are having that bacteria, you are going to change a cheese also. So we are having some video on that, and then you may see. It will show you very good one.
Litmus milk test. The litmus milk test is used to differentiate among microorganisms that enzymatically transform different milk substrates into varied metabolic end products. Litmus milk broth consists of milk, sugar, lactose, pH indicator litmus, and milk protein casein. Depending on the types of enzymes produced by different organisms, a variety of different chemical changes happen in the medium. The pH indicator incorporated in the medium helps in the detection of the production of acid or alkali and oxidation reduction activities of microorganisms. Materials required: tryptic soy broth culture of different microorganisms, inoculating loop, litmus milk broth, Bunsen burner. Procedure: arrange all the cultures and media in the laminar airflow. Take tryptic soy broth from the rack containing 24-hour culture. Take an inoculation loop and sterilize it in the blue flame of the Bunsen burner till red hot and then allow it to cool. Remove the cotton with the small finger of your right hand and flame the mouth of the tube. Take a loop full of culture from the tube. Flame the mouth of the tube again. Remove the cotton with a small finger of...
Do you see that? This one is very interesting if you are going to see this one. Look now. You are going to have already, this is a wrong advice which the laboratorian is giving. When you are doing in laboratory, if you are using right hand, yes, you are going to use the loop on right hand. But this, it says right hand. No, it's not. If you are left hand, yeah, you are holding the loop on left hand. So it makes it different. You see, if you are going to use the left hand, you are using the loop. If you are right hand, yes, on right hand, you are going to use a loop. And always, you remember in the future also, in laboratory, always, if you are having a tube and then there is a lead, the lead must be held by the hand which is holding the loop also. Look at this is a loop. And then by that hand, you hold the lead. The lead mustn't be here. You see, free on this hand. It must be here. So you are going to know two things now. The lead must be already on the hand of the loop holding. And then you can use the loop holding on left hand or right hand. That you need to know it. Okay.
Your right hand and flame the mouth of the tube. Take a loop full of culture from the tube. Flame the mouth of the tube again. Replace the cotton and return it to the rack. Take a sterile litmus milk broth from the rack. Remove the cotton and flame the mouth of the tube. Inoculate a loop full of culture in the broth. Flame the mouth of the tube and place it in the rack. The other 2 test cultures were inoculated successively. Incubate the tubes at 37 degrees Celsius for 24 to 48 hours. After incubation, observe the results. Result: Escherichia coli convert the milk sugar lactose into lactic acid with the help of enzyme beta-galactosidase. The medium turns pink due to curd formation and gas formation. Pseudomonas aeruginosa hydrolyze the milk proteins, primarily casein, into their basic building blocks, namely amino acids. This is accompanied by the evolution of large quantities of ammonia, resulting in an alkaline pH. The reaction is known as peptonization, where brownish supernatant called whey is formed. Bacillus ceres also result in the peptonization of the medium with the formation of rennet curd due to the action of enzyme renin on casein. This forms paracasein, which in the presence of calcium ions is converted to calcium paracaseinate and forms an insoluble clot. Rennet curd is a soft semi-solid clot that will flow slowly when the tube is tilted.
It's very interesting. Now we are coming to the end almost. Look now here, we are going to have MSA. Look here. It's very interesting. You have been doing, look, 3 steps on unknown bacteria, how you need to find the staphylococcus and the streptococcus. 3 steps. Look now. You are going to say staphylococcus is going to have coagulase enzyme, streptococcus, streptokinase enzyme, 1. Secondly, when we were using staphylococcus and then streptococcus, and then when we are using certain media, staphylococcus is going to show you the positive reaction, and then streptococcus, no catalase reaction, number 2. And then number 3, you are going to identify also staphylococcus, streptococcus in a third stage. Look now, it's very interesting. You are going to have MSA media, means mannitol salt agar media. Mannitol salt agar means in that media there is salt in it. If we are going to do in laboratory 0.5, 0.1, and then 1.5, 2% concentration of agar media, we prepare and then pass the bacteria. Staphylococcus, streptococcus. Look now here. Staphylococcus is positive on MS media. Look now. Streptococcus is negative. Positive, positive, and negative. Look now. And mostly, staphylococcus is more dangerous than streptococcus. You see, around, if you are going to keep the food on a table around the kitchen about 4 hours, it is going to contaminate you, food poisoning. You see, that is more dangerous than streptococcus. You see now. So these are very important, especially leave staphylokinase and the streptokinase. In laboratory area, focus it catalase test positive and negative for staphylococcus, and then MS media also positive and negative here now. So we are going to focus on this one. Don't forget it. I'm going to ask you here, the last one. Let's see this one, how it is going to make it.
Another medium we're using to help identify staphylococci is mannitol salt agar, or MSA. This agar has a high salt concentration that inhibits many other bacteria, but not Staphylococci. Staphylococci are able to tolerate fairly high salt concentrations. So we say that mannitol salt agar is selective for Staphylococci. In addition, it contains the sugar alcohol mannitol and the pH indicator phenol red. Phenol red, as we learned earlier, is red at a neutral pH but turns yellow at an acid pH if fermentation of mannitol occurs. So we see that the organism on the left ferments mannitol. It grows on the high salt concentration of the mannitol salt agar, indicating it's probably a Staphylococcus, and also it's fermenting mannitol. The acid end products are turning the phenol red yellow. The organism on the right is also growing on the mannitol salt agar, indicating it's probably a Staphylococcus, but it's not fermenting mannitol. No acids produced, and the phenol red remains red. So the organism on the left ferments mannitol, the organism on the right does not ferment mannitol.
You see, depending on this, to ferment the mannitol, it must have a salt in it. So it is salt-loving type of bacteria, Staphylococcus. So it's very interesting here now. When we are coming to end, let me give you now here an idea, which we say, these all students are going to do for unknown. How many weeks? 3 weeks they do. That means almost in a week, 1 week, 2 days, and then they are going to do 3 weeks. They are making about more than 6 times or more, you see. So when they do this, they are going to find what bacteria it is. Okay, according to the catalase test, and then according to MSA test, what are we going to say? The bacteria is a staphylococcus. Okay, what about spore forming? Maybe it's contaminant, you see, in the beginning, you see. Maybe the two bacteria are different, you see. Sometimes students exchange their bacterial culture and then they are going to use it. The one which is in gram stain, gram positive, and then spore forming bacteria mustn't come to staphylococcus. So there is some kind of mistake is done, you see. So for this reason, you don't need to take somebody's culture. You need to work with own culture. But here in this case, because of MSA agar and then catalase test, the student is having a staphylococcus. Now, the student is going to conclude. For example, Dr. Asi wants about 2-page explanation. I don't need it. I want only the short answer and then finish the activity. 5-page, what they have done, plus they need to give me conclusion, this answer. So when they conclude, they say, the strongest indicators were the staining test that revealed gram-positive spore-forming non-acid-fast bacteria. You see that? They say spore-forming. Now, Staphylococcus is not spore-forming. You see, there is a mistake already. Now, so for this reason, I need to go one by one the step and then I see it. You see, where the student have done mistake. Based on the cultural, morphological, and physiological tests, you see fermentation processes, which you have done, test, and utilization of Burgess manual classification, I can conclude that the unknown bacterium is Staphylococcus. Well, what I may do, this student have done all reaction. And then if that student miss only by naming this student, he's not going to lose so many. Maybe he is going to get 60 out of 80. Because all the reactions I will go and then see it. Or because he has done all of them, because he said spore, maybe I may take 10 points from that. And then maybe 60 or 70 out of 80 will get that student. So this student has done his best. So only by naming the bacteria, I make a mistake. So for this mistake, I'm not going to give for that student 50% or whatsoever. No. So the student has done it. Because of that activity, I'm going to give that student maybe 70 out of 80. You see, that is how we are going to find unknown bacteria. Look, very interesting. For this reason, what you need to focus on for your final exam. I will check over this one. After 1 or 2 hours, I will send you this information. You keep it. Okay, now, let me give you some information which you need to do. One, the evening student on the lab exam, you have done better than the morning students. But there is one student from the evening didn't take the exam, and then from the morning, 3 students, they didn't take the exam, and they didn't inform me also. If they don't have a good reason, I'm not going to give them the exam. You see, they are going to get 0 for that. You see, now for that, we don't discuss on the exam until they take. Okay, now, the second one, which if there is something you want explanation by yourself, email me. I will respond it to you. Okay, now, so we need to wait. The second one is about your lecture exam. Lecture exam, you have about 406 questions. Okay, now, I will give you a word now. Remove those true false questions. Most of them, I think they are about 40 maybe. I am not going to ask you from true and false questions. Eliminate them. So matching questions, I will ask you. So true, false, you don't need to study. Okay, I will write that one on announcement also. Okay, now, reading about lecture exam, I will wait really. Most focusing on the lab exam. You need to focus lab exam. You need to read more on lab exam, even though it is given lastly. So you need to focus more on lab exam. So that lab exam information, I will send you tonight. You are going to get it. Thirdly, the lab activity from lab manual, which I send you on September 2, I posted already a rubric, how you need to answer it. Rubric there. So how you need to get points on that rubric, I put it. So it is going to be open on Monday on 16th of November. So on begin Monday up to December 10, you can submit anytime, you see. So the information, how to submit, it is written there, you see. If you are having a question, you email me. I will answer it, and then I will explain it to you. So for this reason, try to focus how to answer it, you see. In instruction, rubric, you need to read the rubric, you see. What the rubric says, follow it. That is your instruction how to submit it. Very, very important there, you see. So in the site of discussion, we are going to submit. It is there also. It is easy to submit. No problem with that. So here, what we need to do, some of you, you didn't buy the book, but if there is somebody who has it, you contact them and then let them send you the page and then get from them and then answer it, you see. You don't need all of you to buy it. So get them and then get that information, answer it on that paper and then send it. Not on the paper, on the other paper. Please communicate with someone, and then someone who bought already that page, and then that page, if you copy, and then you may exchange with someone and then answer it on those pages. And then I ask you only certain page, not all page must be sent. So that's why you need to follow the rubric, you see. So the rubric is giving you explanation of everything to do. And that is very important. The fourth or the fifth thing which I need to mention is really this class, last time, the morning student, 10 students, they didn't do activity. 4 points. Because it is 4 points, they didn't want to do 10 students. But your class, no one is missed. No one missed that activity. All of you have done it. That's very important. Anything, any droplet, any drop of points which you get, it is helpful for you, really. On today's activity, it is 6 points also. Really, you need to go Google. You can have a book. You can search it anywhere else and then send it. If you are going to get it from the Google area, change it somehow and then submit it. Really, I don't need references. Without reference, you can submit it, please. I didn't see one student who sent me that report on viruses, really, on 4 viruses, how they answered. You see how the students are serious. Some students, they write about sentences, even they don't correct their sentences. Their English is worse than mine, really. They write it and then they don't make capital letters, and then because I say sentences, they cut it off and then they leave it as it is. No, it's not. Before you do it, check, please. I gave you some, and then you need to choose only certain of them. And then you need to answer, not all of them, you see. So for this reason, please, when you see it, read the question and then answer it. And then don't miss it. Also, there is one student from the morning student, he said he is missing for 4 minutes and then he didn't submit it. And then he asked me, can I submit it? I said, no, the time is past, you see. If the time is past, I'm giving you more than 2 hours. That will take you about 30 minutes, really. Really to do it, 30 minutes, it will be enough. But there are certain students, they don't do it, and then maybe some students are not present in the class, and then finally they want to submit it finally. And then that I know it, you see. For example, morning, I have 41 students, only 33 students were present in the class. Look now, those are students who are trying to submit lately, but I gave them the hour to submit up to, not up to the end of the class, but I make it shorter, you see. So for this reason, read it at what time you are going to submit, up to what time you are going to submit, see it and then answer it, really. It's not hard, really. I admire you, how you have done it. Especially, I'm very thankful for the student who have got on this exam above 88, really. Above 88, you have done very good. But there are students, about 7, 8 students got 100% also. I'm proud of you, really. I'm proud of you. You can make it, really. It's beautiful. It is guaranteed. When you do this one, it is guaranteed that you are almost the highest rank performing students, really. Very good. I'm very happy, especially 100% getting students. I'm telling you, if there is a laboratory, I want to work with you, I will work. Really, I admire you. There are certain questions are very critical. How you answer it, it's amazing. I'm very proud for those students. If you want, some of you have got somehow low grade, it doesn't matter. It doesn't matter. Work on this exams, and then bring it. Activity, don't miss it. And then lab report, which are going to present 80 points, don't miss it. Please follow. Help each other and then submit it. That is all what I'm going to say. At 7 o'clock, your activity begins. So is there any question you want to ask me before you go to your activity? Almost there are. You don't need to write your name, or you don't need to say at this time, I recorded already. Thank you very much, really. I recorded today. This system, it records your name, and then it will send me at the end of the class already, you are present or not. So I'm very thankful. You are almost 40, 41 students in the class. Here I see about 40. It's good enough. You see, it's good enough. 40 students. 39 students is good. 38 is good. You see, some students, some problem exists. They miss something. I'm going to send you. You will read this one, and then you are going to get it. You see, so I'm happy. Really, I'm happy. And then that is all. And then for those students who are taking final exams, look now, when I give you the lecture exam and then lab exam, I gave you a gap. So what I may do is, on a gap for lab exam, I'm going to review with you guys. Okay, now, that is good one. Okay, and then the ones who are taking physiology with me, I gave you a gap, you see. You are taking exam on the 10th, and then the exam in microbiology is on the 8th, you see now. I gave you a gap between. And then I'm going the exam early. One of the exam I gave you early on 4th, you see, 4th of December for physiology students. So it will help you to prepare only for the one exam, which you are going to take 10 also. So I think already, and then that information is sent to the college also. In a college syllabus, I send it. So the academic dean, our vice president, know my syllabus. So I cannot change it. That syllabus, you see, it is submitted already. So for this reason, try to work and then make it best. If you are having certain questions, you get some problem exists, please send it on Canvas site. Please don't email me on the other email site. Send me on Canvas, I will answer it. That is very good, and then operation. And I appreciate that too. Thank you. And then I wish you a good weekend. Be careful. COVID is increasing. I told you from the beginning, COVID is going to increase. Now, myself, I don't want to run to the markets really. Almost, maybe in 3 weeks, one time, maybe. For this reason, please, your children, older people in the family, you need to take care of them, please. If you are having, for example, there is a rule come. Do you know what they say? If you are working in some kind of business, for example, I have a business with my wife, and then they are saying you need to be checked 2 times a week. How it can be happen? Do you believe that? I don't. I don't think I'm going to do it. But not to lose my business or our business, maybe I will be checked, you see. But the only thing is, maybe the people who are going to work, I need to reduce it. Reduce the people who are coming from outside, and then we will take care of it, that business, you see. So be careful when you are working with someone, everything, please use the mask. If there is possibility, N95. Hey, another thing which I am going to appreciate you. You remember, I posted for you, the college is having money and then giving you. And then I ask you, use my name and then ask them. There are 5 students already from micro morning and evening, they are granted money. It's a free money. Look now. How many of you tried that? I gave you the chance. You need to apply and then maybe you may get the money. You see, if there is opening there, please go read that one and then apply again, maybe. So 5 students got the money already. They are going to get it. So I approved the names that the students are enrolled in my class. You see, I feed you, I gave you that announcement, and then they applied, and then they got it, you see. Look now. So for this, why you miss? Now today, they advertise also, there is a food weekly base. They are having excess food in the campus pantry. If you are going to look for food, please contact the college. There is food extra, you know. So they are going to donate, you see. When I was distinguished faculty and then get that $500 they pay me, I gave for that book that food pantry also for the people who are in need of food that the college may buy and then provide them, you see. So for this reason, there is food. If there is food, contact them and then go and get it from the campus. Why not? If it is free, you see. So if I were in somewhere else, I don't have money, I will do it. If there is less money, I will do it. It's a free money. And then around Thanksgiving, they are going to give turkey also. You be ready. You go and then get it. Why not? It is not a very long line. Go and then get it for your family. If there is somebody from your family, take someone together also. They can give them. They are, yeah, you can go 3, 4 people, you go, you can have turkey. You see. So why? Why not? Really. About the money, check again. If that is opening is still there or not. Try it. If there is a way you can apply it, then apply it. I will submit your name. You see, they are going to ask me if these students are present in your class or not. So I will do that. So go on. That is all I have. I'm very thankful for you. And then see you, and then go do your job, and then meet you again. If there is any questions there, I will answer it there. I'll wait. You see, good luck. There are some students already left while I'm talking. So I wish you the best and see you again. I don't want I have family members who are positive for COVID. Well, they are doing very well now. And we are working with them, you see. It's good. They are hopeful to be cleared, you see. About the vaccine which is coming now, if I were you, I'll take the vaccine, you see. I will just check yourself. About 90% is successful. So are you going to use it? Welcome. I'm going to take it. I'm going to